Hi everyone, we continue to learn about the analysis of proteins. Elucidation of the three-dimensional structure of protein is often the source of a tremendous amount of insight into its corresponding function, inasmuch as the specificity of active sites and binding sites is defined by the precise atom arrangement within those regions. X-ray crystallography and nuclear magnetic resonance, short for NMR spectrophometry, are the two most important techniques for elucidating the conformation of proteins. X-ray crystallography was the first method developed to determine protein structure in atom detail. This technique provides the clearest visualization of the precise three-dimensional position of most atoms within a protein. Of all forms of radiation, X-rays provide the best resolution for the determination of molecule structures because their wavelength approximately corresponds to the length of a covalent bond. The three components in an X-ray crystallography analysis are a protein crystal, a source of X-rays, and a detector. When a narrow beam of X-rays is directed at the protein crystal, most of the beam passes directly through the crystal, while a small part is scattered in various directions. These scattered or diffractionated X-rays can be detected by X-ray film or by a solid-state electronic detector. The scattering pattern provides abundant information about protein structure. So, as shown in the figures, the X-ray photograph is a two-dimensional section through a three-dimensional array of 72,000 reflections. The intensity and the positions of these reflections are the basic experimental data of an X-ray crystallography analysis. Each reflection is formed from a wave with an amplitude proportional to the square root of the observed intensity of the spot. So each wave also has a phase, that is, the timing of its crest and throws relative to those of the waves. The next step is to reconstruct an image of the protein from the observed reflections. The image is formed by applying a mathematical relation called the Fourier transform to the measured amplitudes and calculated phase of every observed reflection. The image obtained is referred to as the electron density map. It is a three-dimensional graphic presentation of where the electrons are most densely localized and is used to determine the position of the atoms in the crystallized molecule. X-ray crystallography is the most powerful method for determining protein structures. However, some proteins do not readily crystallize. Then nuclear magnetic resonance, like NMR spectroscopy, is unique in being able to reveal the atomic structure of macromolecules in solution, provided that highly concentrated solution can be obtained. This technique depends on the fact that certain atom nuclei are intrinsically magnetic. Only a limited number of isotopes display this property, called spin, and those most important to biochemistry are listed in this table. So the simplest example is the hydrogen nucleus, which is a proton. The spinning of a proton generates a magnetic moment. This moment can take either of two orientations or spin states we call alpha or beta when an external magnetic field is applied. A spinning proton in alpha state can be raised to an excited state like beta state by applying a pulse of electromagnetic radiations, provided that the frequency corresponded to the energy difference between the alpha and beta states. In this circumstance, the spin will change from alpha to beta. In other words, resonance will be obtained. So these properties can be used to examine the chemical surroundings of the hydrogen nucleus, the flow of electrons around a magnetic nucleus generates a small local magnetic field that opposites to the applied field. A resonance spectrum for a molecule is obtained by keeping the magnetic field constant and varying the frequency of the electromagnetic radiation. The different frequencies, termed like chemical shifts, are expressed in fractional units d, short for per million, or ppm, relative to the shifts of the standard compound, such as water-soluble derivatives of a tetramer silicon, and that is added to the sample. For example, a CH3 proton typically exhibits a chemical shift d of 1 ppm, compared with a chemical shift of 7 ppm for an aromatic proton. And the chemical shifts of most protons in protein molecules fall between 0 and 9 ppm. So with this information, we can then deduce changes to a particular chemical group under different conditions, such as the conformational change of protein from a disordered structure to an alpha helix in response to a change in pH. Then we can garner even more information by examining how these spins on different protons affect their neighbors. By inducing a transient magnetization in a sample through an application of a radio frequency pulse, we can alter the spin on one nucleus and examine the effect on the spin of a neighboring nucleus, especially revealing a two-dimensional spectrum obtained by nuclear overholster enhancement spectroscopy, so short for NOESY, NOESY, which graphically displays pairs of protons that are in close proximity, even if they are not close together in the primary structure. For example, as shown in this figure A, is a schematic representation of a polypeptide chain highlighting 5 particular protons. Protons 2 and 5 are in close proximity, whereas other pairs are further apart. And in figure B, it is a highly simplified NOESY spectrum of a peptide. The diagonal shows 5 peaks corresponding to the 5 protons in part A. The peak above the diagonal and the symmetric related one below reveal that proton 2 is close to proton 5. So here is a two-dimensional NOESY spectrum for a protein comprising 55 amino acids, as shown here, and the large number of diagonal peaks reveals short proton-proton distance. The three-dimensional structure of protein can be reconstructed with the use of such proximity relations shown in the NOESY spectrum. The structures are calculated such that protons that must be separated by less than 5 angstrom on the base of NOESY spectra are close to one another in the three-dimensional structure. So if a sufficient number of distance constraints are applied, the three-dimensional structure can nearly be determined uniquely. So here is for today, and thank you for your attention.